Welcome, good afternoon, everyone, and to our third of five in our October and Beyond series. In this November lung month that we were all just discussing, we were blessed with beautiful weather. So glad you could join us today. Our theme today is really around the evidence-based in our efforts for occupational disease prevention. So I am going to pass over now to our COO, Dave Wilkin, to introduce our first speaker for today. We just logistic-wise, you can put questions in the chat window and we'll have time for Q&A after each speaker. We really are happy to have you and hope you can join us for the next two sessions in the next 2 Fridays as well. And you'll see lots of helpful links in the chat window as we proceed. So over to you, Dave Wilkin. Thank you.
Okay, thanks. Good afternoon, everyone. Our first presenter is Professor Barbara Hales, speaking on the topic of endocrine disrupting chemicals in the workplace. OCA has been in touch with her and her colleague, Professor Bernard Robert, as part of our cluster investigation work on the Pebra, now Ventra plastics plant in Peterborough. And we thank them both very much for their work in helping to educate our staff on this topic. Dr. Hales is a James McGill Professor in the Department of Pharmacology and Therapeutics at McGill University, a past president of the Society of Toxicology of Canada and the Teratology Society, now known as the Society for Birth Defects Research and Prevention. She's co-chair of the Government of Canada Chemicals Management Plan Science Committee and Secretary General of the International Union of Toxicology Societies. Her group's research is focused on understanding how chemical exposures adversely affect reproduction and development. Projects in her lab funded by the Canadian Institutes of Health Research include the effects of house dust mixtures of flame retardants on reproduction and development, the impact of exposure to phthalates and green plasticizers on progeny outcome, and approaches toward the responsible replacement of endocrine disrupting chemicals. And with that, I will hand it over to Professor Barbara Hales. Thank you for being with us today.
Thank you very much, Dave. Great. Okay, so I feel a little bit guilty because I was co-chair of the Science Committee for the Chemicals Management Plan and I was the Secretary General, but I'm not there anymore. However, we still are very much interested in endocrine disrupting chemicals and learning about them in the workplace, as we found out when we started talking to Dave and his colleagues, is that there's less known than you would hope in this area. So what do we know? My topic outline is here. So first of all, what is an endocrine disrupting chemical? We're going to talk a little bit about what we know about environmental exposure to these chemicals because there's much more data. A little bit about what we know about exposures in the workplace, and then future directions and challenges. So first of all, what is an endocrine disrupting chemical? So the widely accepted definition is the WHO definition, which is that an endocrine disruptor is an exogenous substance or mixture that alters functions of the endocrine system and consequently causes adverse health effects associated with exposure. This can be in an intact organism or its progeny or in subpopulations. So if we think about how these chemicals might work, on the left you have normal hormone receptor and response. The endocrine disrupting chemical could be a hormone mimic, in other words, activate the same receptor as the normal hormone, or it could be an antagonist or blocker of the response to that hormone. So it could also cause, as we see on the bottom as well, a direct effect, either stimulation or inhibition of the endocrine system, or the overproduction or underproduction of the hormones that are of interest or concerning. So if we look about, okay, the endocrine system is really sort of the internet or the master control board for the body. So there's actually receptors, hormone responses in most tissues in the brain. the brain, the hypothalamus, the pituitary in bones, mammary glands, uterus, oviduct, prostate, in fat cells. It's very important in the adrenal and the heart. So basically, many organs in the body can be affected by these potential endocrine disrupting chemicals. What can be the consequence? So what kind of impact could they have in terms of adverse health? So if we have these impact, we can sort of go around the circle and we can see that there could be metabolic disorders, for example, diabetes. We think always, I guess, when we think of endocrine about reproductive effects, but there's also immune function and disease. Thyroid is an endocrine tissue. Bone, there's receptors, steroid hormone receptors in bone. Neurodevelopment, a number of these chemicals have been associated with developmental effects in terms of behavior in children, and hormone-related cancers. So there's a wide variety of possible outcomes as a result of exposure to an endocrine disrupting chemical. And the other kinds of additional considerations. So what you see is not necessarily an acute result. It could be a result of a long-term exposure if we start over on the left. So the exposure could have happened over a lifetime. It could have happened decades before you actually see the adverse health effect. Real-life exposures are often observed after exposure at sensitive life stages. And of concern, and we'll go into why, is that some of the exposures could be low dose, and the response curves are not always what you would expect, and I'll show you some examples. And then we mentioned before mixtures. So many of these classes of chemicals don't come as just one specific chemical. There's actually families of chemicals that could be interacting in different ways. So there are other effects. So if we look at real-life long-term exposures, one example of a kind of exposure that we may not associate with a specific exposure that happened, last week or last month or last year, is perhaps an exposure to some of these endocrine disrupting chemicals in pregnancy. Yes, there's an effect on childhood, but there could also be lifelong effects on puberty and reproduction in the progeny. Some of these outcomes we now know are only observed when the child has grown up and is now in their 40s or 50s or older, and even in later life. So there are long-term consequences to some of the exposures of these endocrine disrupting chemicals. Dose response curves. We mentioned that it's not always very as straightforward as you'd like. So what you can see is that there is a response, increased dose, increased response. That's classically what we'd expect. But there's many shapes of these dose response curves, and so it's not always evident that increased exposure is going to produce an increased response. They could be U-shaped, depending on how you look at it, upside down or not, and there could be a variety of other dose-response relationships. And this makes it specifically hard for people to assess the association between exposures and health outcomes, because sometimes it's not directly related to the dose that was received. And this just makes it even more evident if I show you a U-shaped dose response curve. So classically in toxicology, we sort of look for specific things like the low L, or the lowest observed adverse effect level. We looked for a response where there's no effect, and we try to define what is safe based on those. So that here, maybe this would be a safe dose, but then, this dose response curve is U-shaped. So what happens up here? And there could be multiple explanations for this, but this makes it very complicated in terms of figuring out what is causing what, or what is associated with what. And just as an example, I'm going to use bisphenol A, and maybe this is just to show you that there's different targets and different reasons for this. So bisphenol A, it's an epoxy resin. We're going to come back to it later in the talk. But very low dose exposure to A could activate specific receptors, hormone receptors. And you can see here that the hormone receptor that's being activated is estrogen receptor beta, or ER beta. At higher doses, 100 times higher, Maybe what you're going to do is actually affect different pathways. So here we have the estrogen receptor alpha being activated, and the dose response curves to these different hormone receptor activations actually are different, so that you're getting, when you combine them, you're getting one of those weird U-shaped dose response curves that I showed you. So parallel but different mechanisms of action could be the explanation for this. And now we know that it's not even as straightforward as this slide makes it seem, because there are also other flavors of estrogen receptors. So that sort of is a very quick overview of what is an endocrine disrupting chemical and the spectrum of the types of effects that might result and the complexity of assessing the association. So what do they look like? What are we actually talking about? Which endocrine disrupting chemicals are found in the environment? This again is a slide from WHO, just to show you that these chemicals can be found basically everywhere in our everyday life. They're in the computer that you're all sitting in front of, or your cell phone that's, I'm sure, very attached to you. They could be in our water, in food, definitely in air, cosmetics, which some of us wear, cleaning products, okay, and other ways, clothing, and in this example, upholstery. The kinds of chemicals could have different functions. They could be used as flame retardants. Some of them are metals. You're interested in plastics and plastic drugs. So some of the drugs that people take end up in water, in the city water, and in drinking water. Pesticides, herbicides, fungicides, polycyclic aromatic hydrocarbons. We talked about bisphenol A briefly. And so this is all kinds of different chemicals with basically sources all around us. So I'm going to give you a few examples of some of them. So when we talk about polycarbonate plastics and epoxy resins, bisphenol A comes to mind. The plasticizers, classically phthalates and related compounds, and sort of the prototype chemical could be DEHP. Flame retardants, some of these were probably brominated diphenyl ethers, which have now been regulated. I'm going to come to that. Organophosphate esters have been replacing them. We probably have all read somewhere about the perfluorinated chemicals called forever chemicals that are in things like Teflon, other pollutants. DDT is the classical endocrine disrupting chemical, and then some of the metals or leads. So what I'm going to tell you about, because I can't talk about everything, is really one story, and it's on bisphenol A. So bisphenol A, it's a huge market. So the global consumption in 2015 was estimated at 7.7 million, and it's projected to be increased to 10.6 million in 2022. It's found in our plastic water bottles, or it was, children's toys, electrical wires, automobile parts, tableware, medical devices, sports equipment, food packaging, and this is a prime source of interest to many researchers. So basically, there's many products that have bisphenol A or something functionally equivalent, and it was before this. Now it's being gradually replaced, but it was bisphenol A. So this is just for your interest and reference. This is an example of a safety data sheet for bisphenol A. And I've cut out a lot of the information. I just really kept the part of the toxicity part. And endocrine disruption does appear. It says no information available. Many of these other things like birth defects, teratogenicity, developmental effects, reproductive effects, mutagenic effects, no information available. And we actually, this date is 2018. And there was information available in 2018. There was information available for many of these topics a number of years before that. But the safety data sheet does not list it or does not. And the bottom line at the bottom of the table is other adverse effects. The toxicological properties have not been fully investigated. So it's not that easy necessarily, unless people go to the primary literature, to find out what what stories or what data are with respect to each of these potentially adverse effects. So I'm going to show you one graph. So this is just a little bit, and this is not that recent. Things have changed, but this makes the point very dramatically, where they've shown us the mean bisphenol A concentrations in relation to reported diseases and conditions in the general population. And what you can see is that there's a long list of conditions from arthritis, cancer, cardiovascular diseases, diabetes, liver disease, respiratory, bronchitis, stroke, thyroid, so many different potentially affected tissues or organs. And if you look at the diagnosis, whether the condition was reported or not reported relative to the mean BPA level, you find this whole box in the middle where there was cardiovascular disease, cancer, etc., diabetes reported in the higher exposed group. So these people were exposed to higher levels of bisphenol A than the ones where the condition was not reported. There's other diseases and effects where that's not true, and any effect on reproduction is not in this graph because the data were more confusing. So as a consequence of many, many studies, so the data I showed you were from back in 2008 and many other data, BPA has been assessed in Canada, and this is a timeline of the BPA regulations in Canada. So in 1996, the Food Directorate established a provisional tolerated daily intake of 25 micrograms per kilogram per body weight per day for bisphenol A. In 2008, the Government of Canada released its final report on the health risk assessment of BPA, and they've updated it since then. But as a consequence of this 2008 report, by 2010, the manufacture, importation, sale, and advertisement of polycarbonate baby bottles containing BPA was prohibited, and also it was later prohibited in cosmetic products. Well, what about in the European Union? So in the European, the first full risk assessment of BPA came up with a higher tolerated daily intakes. This was 2006. And then we go through, and many of the different countries, you see that regulated it separately, so that in France, Sweden, by 2012, a lot of focus on food packaging and children, food packaging material here too, and children. There was a comprehensive review, and there the tolerated daily intake was brought down to 4 micrograms per kilogram body weight per day. And if we walk over to 2018, again, there's a lot of regulations in terms of the use of BPA in a variety of products, not just children's products. So the consequence of regulations is that you have to replace the chemical, right? If you're not, if you still need the functions of the product, you have to find something else, some other epoxy resin or some other equivalent chemical to replace it. And the same thing is true for flame retardants. So unless the regulations are changed in terms of flammability, you need a flame retardant in the product. So people have, as things got regulated, like the brominated flame retardants, the PBDEs, they've been replacing them with organophosphate esters. As plasticizers like DEHP are regulated, they move to longer plasticizers, other phthalates, sometimes other products, but there's still a plasticizer in your medical tubing and in many products. And epoxy resins are an interesting story because the migration has been from BPA 2, and you can see the long list, and the list is huge. So BPS, BPF, BPAF, and we often don't know which of these chemicals is in the food packaging or the baby products or the different cosmetics that we use because of this alphabet soup. And recently, Health Canada, the Chemicals Management Plan, looked at what kinds of bisphenols and tried to put them together. I think their list had over 300 different bisphenols. So this is just a glimpse of what might be going on. So the team grant that Dave mentioned that we've been working on is interested in these 3 categories of chemicals. And we're interested in specifically what is replacing the legacy chemicals and whether whatever is replacing them is safer than the legacy chemical that was regulated. So basically what I'm saying is that if you regulate something, there's a market gap and there are replacements. So the question that our team posed was, is industry shifting to safer alternatives? So i.e., is it regrettable substitution or responsible replacement is the other possibility. And so obviously we all hope that it's responsible replacement. Sometimes it's not the regulations that are the pressure. So sometimes it's public pressure. So with all the publicity on BPA, the public actually probably had more influence than government regulations across the world. So the public wanted BPA-free products. And so the interest really is in, are they healthier? So are these alternatives, as some of these headlines show you, are these alternatives also potentially endocrine disrupting chemicals? And so that's a question that we don't know really, because many of these different bisphenols we don't know very much about. So what I'm going to tell you about is just a little bit of a story about relatively few major bisphenol replacements. So this is Bisphenol A. So what you can see from the structures is that some of these, Bisphenol S and Bisphenol F, are actually very similar. They're not so different from Bisphenol A. So if we look at human biomonitoring, and in Canada, the Canadian Households Measure Survey monitors Bisphenol A, but we don't have the data for the other bisphenols at this time. So I'm showing you data from US children in the NHANES data, and this was a little while ago. So if we look at BPA, you can see that, you know, we have family, age, everything, a lot of information about who was exposed to what in terms of BPA. BPF, people were exposed to it all way back in 2013, 2014, when Bisphenol A was still being regulated in a number of countries, and BPS. So we now have BPS. So exposure's happening. Just because we don't have the data in Canada doesn't mean that we don't have BPF and BPS in our bodies. So what about the workplace? So there's almost no information about exposure to bisphenol A or these different bisphenols in the workplace. There was so what I'm going to tell you about is based on a systematic review that was published only this year, where they were putting together and reviewing all the studies that they found in the literature that monitored occupational exposure to BPA, BPS, and BPF. And it's only these three bisphenols because there's basically no data on any of the others. And even if you look at the color code, BPA, there's quite a few studies. BPS, BPF, there's very few studies, and only in actually 3 jurisdictions, so China, US, and Spain. And you can notice if you look at the bottom, there's no Canada here because there were no studies done in terms of biomonitoring of this in Canada that were in an occupational exposure setting. So what conclusions did they come up with, or what is the basic of? So the industry, the occupations that they found these papers were based in were obviously the manufacture of these epoxy resins and their use, molding polycarbonate plastic, shipyard workers, and this was partly in the paint that the shipyard workers were using, market sellers, cashiers, petrochemical company, healthcare workers, teachers, office workers, salesperson, firefighters. So a wide variety of different occupational settings. Only 4 of these studies, there were about 30 there, had female subjects, so they were mostly men. Some of the studies did not have a control group. When they did have a control group, the difference between the workers' data and the control data were significant. There were different ways of collecting the data. So in some cases, they were often doing pre-shift measurements, and there were some other strategies too. So in terms of effects, difficult, because most of these studies were quite small. There were lower serum hormone levels, testosterone levels, with increased BPA, associated with increased BPA in some studies. And some of the studies have reported an association with effects on sperm quality. That's about it. If we look at a sort of a summary of these occupational human biomonitoring studies, you can see that the blue again is BPA. We have a little bit of BPS and BPF, and these are in Europe, and the rest are mainly in Asia. They highlighted incinerator workers for the BPF exposures, cashiers for the some of the exposures to BPS, and a lot of plastic epoxy resin production, and using basically the raw BPA and being exposed to that. They highlighted also the need for new studies on bisphenol substitutes, BPA substitutes, to identify tasks with high exposure and to have some kind of harmonized regulatory approach. So there are many countries where BPA is not regulated. I showed you some where it was regulated, and more information on the mechanisms of action. Because if you know what mechanisms to look for, then maybe you don't have to do as extensive studies with this 300 odd bisphenols that I mentioned, that you could actually target a mechanism. More on exposure, so that we can figure out how to avoid it and possible health effects. So if we come to future directions and challenges, we'd like to predict exposure. There's lots of programs now and lots of databases, and EPA has developed quite a few. I'm just going to tell you about one of all these, a high throughput cell culture assay that we've been doing. So this is a CIHR funded team grant. There's lots of people involved. So we have one aim to measure exposure in food and in water especially, and also in breast milk. One to look at bioactivity, and that's with Dr. Bernard Robert, and I'm going to tell you a bit about those data. And then we're working with Jay Ellis, who's a lawyer, and other people to really work on promoting responsible replacement policies. So the bisphenols that we looked at were these ones. So we had Bisphenol A in blue, and then some of the others I've already mentioned, and a few others. Specifically, the last two, the BPTMC and BPM, were suggested to us by colleagues in Health Canada because there's really no data, and yet we now are learning that we are exposed to these chemicals. So we did cell cultures. So we basically put cells in a dish, we put in some fluorescent dyes, and then we look at them with a high content imaging system to see what's happening to the phenotypes of these cells after they're exposed to bisphenol A. We can do some manipulations to be able to relate the concentrations in a dish to those that people might be exposed to. So this shows you some of the pretty pictures. So we can see that we can have big increases in going from control to different. This is bisphenol A, this is BPTMC, this is BPF. So we can see different, basically pretty pictures to show you that, oh boy, there's not much fluorescence in this one, but there's tons of fluorescence in this image. So that the exposure to these bisphenols at these concentrations can increase the amount of response we have to these different markers. So this is actually an increase in lipid droplets, which could reflect an effect on steroid production. So we can see if we look at the red bar, BPM, there's a big increase at 20 micromolar in lipid droplets, and it doesn't happen at the same concentration for BPA. You need to go up to 100. So some of these chemicals are actually more potent than the legacy chemical that they are replacing. And we can show this here where we look at effects of many of these chemicals, and we see that they happen on all these different markers, lysosomes, mitochondria, lipid droplets, and oxidative stress at concentrations that are often below those of BPA. So the question is, can we define a safe exposure level? If we do, usually that assumes that the dose response curve is monotonic, but that's not always true from what I've showed you. What happens when we're exposed to a mixture, because these bisphenols often are found in our bodies, and they're not found just alone. When you measure these different ones, many of them are there. Can we look at study designs that can accommodate effects that are delayed? So we don't expect to see necessarily a response immediately. It might take decades. And what are the criteria that we can use to regulate these kinds of chemicals? So just to show you an example from Europe, because it's complicated, is there's all these different legislative jurisdictions to regulate the different chemicals that people are exposed to. And criteria exist for identifying endocrine disrupting chemicals only for certain classes of these chemicals. Many of them do not have criteria in terms of how to evaluate these chemicals. So in workplace safety, there's not actually a class for most in terms of endocrine disrupting chemical in the labeling of chemicals in the global harmonized system of classification. So labeling is an issue. We already mentioned the lack of coherence across regulatory sectors. So you may identify something as an EDC in food contact, but not in medical devices. And the difficulty in risk management when you don't know the types of dose-response relationships. So I just wanted to briefly tell you that lots of people have been involved in these studies in university settings in Canada and in South Africa and in Health Canada. And that was the end.
Thanks, Dr. Hales, for all of that amazing presentation. I know you've spoken to some of our group and you will be speaking again for any questions in the chat window. I did just want to again commend you for pushing everything into a smaller time frame. I know we were we were hoping we'll have additional opportunities in the winter webinar series, hopefully to expand further and on some of the research. And it's really great to get refreshed on toxicology again and why it's important and what we do in terms of occupational disease prevention, and remind ourselves that this evidence is continuously changing, and we just keep producing things and substances, and we don't know when we're trying to replace, as you spoke about, what the health effects might be of those. So any questions are welcome in the chat window. Thanks, Paul, for your commentary. For Dr. Hales, I'm not sure I'm seeing any immediately. Everybody should get up and do a stretch while we're while you're thinking of questions, so that you're not overly WebExed out or zoomed out. Paul's thanking you especially for the responsible replacement, offering us a great challenge to our point about how do we do that when we just keep, like I said, creating substances, hoping that they have lesser health effects than others. So it is, and global harmonization, we think, helps with all this, but you point out, the weaknesses in a lot of that. So, it's a challenge, but it is good to be aware and to know about those. Sarah Miller is asking, oh, okay. Sarah, can we cut to the chase and get reliable and recommendations for healthier? Yeah, go for it, Dr. Hales.
So I think that's one of the major goals of some of the Health Canada people in terms of communicating to people what is known. It's really complicated, Sarah. Great question, but no simple answer for sure.
Yeah, and stay tuned, Sarah. I think the Health Canada folks are coming to the November 26th session, and they are going to be speaking closely more around the flame retardants, specifically with the chemical management plan that they're working on. And next week, Kevin is introducing Dr. Thomas Tenkeg. He's talking about some control banding we can do to help with these recommendations, and again, learn from the work that Dr. Hales is presenting, and realizing that we may have to categorize things into how they work and what they look like in order to come up with healthier recommendations while we wait on data sometimes. So yeah, good question. Any other questions for Dr. Hales? A couple coming in here. Seems like a daunting task, but is there a positive future in the control of these chemicals?
We certainly hope so. We're spending a lot of effort to look at what's in our environment, not occupationally, but in our environment in Canada and in South Africa to try to compare them. And we're working very closely with regulators, and obviously regulators in Canada, with the Department of the Environment in South Africa and a number of people in Europe. So there's a lot of conversation going on. There's obviously many more questions than answers, but we are measuring a number of these bisphenols now in products, in food, in water, and in breast milk in Canada. The data are not out yet.
And yeah, routes of exposure can become important as well, right? And different, we talked combustion products, we talked chemical reactive products, we talked raw product, we talked product in materials and products themselves. So Kevin, why don't you go ahead? You have a question. I see your hand up.
Yeah, sure. Thanks, Dr. Hales. It's really interesting because as part of a project that we've been working on for Health Canada, one of the things we've been looking at is BPA in thermal paper handled by cashiers. And it's kind of been a bit of an eye-opening exercise for me to look at all these cashiers that are exposed to BPA through skin contact. And, you know, I kind of read a lot on that and sort of learned a lot about it. But I'm just wondering, are you aware of any focus in Canada on that, cashiers' exposures to BPA?
I'm not sure. You know, I know that people are also interested in BPS and exposure of cashiers to BPS. I think a lot of this research has been happening not in Canada, but in the US. So it's not just BPA, it's BPS. And I think, again, this is one of those examples where maybe the public is more aware than maybe the researchers sometimes, because I know, you know, now cashiers everywhere say, do you want your receipt? And, you know, many people say no because they know they don't want it. They don't want to touch it. Or, oh, should I put it in the bag for you instead of handing it to you? So I think there's an awareness, but I don't think we have all the data for sure about what's in there.
Yeah, and I guess the other issue is that because it's an article, you know, there's no safety data sheet, so to speak, for thermal paper. So cashiers don't actually fully understand that they're being exposed through skin contact.
I think the data sheets are incomplete, and I don't know, you know, that it's very hard to get them to be updated. And unless you call something, you know, endocrine disrupting doesn't hit high flags for people, so they don't really worry about it. It's often ignored because the toxicity tests are complicated to measure endocrine disruption. For the examples that I showed you, there's so many endpoints.
Thanks so much. I love the great presentation.
Thank you, Dr. Hales. And as we don't see any further questions popping out, I think we'll thank you again for joining us. And a reminder that this is recorded and that we will be having you folks back and to get updates on a lot of this research that the group is working on. And I know there's a couple of your colleagues that wanted to share some of their current very close to being published info. So we'll have those colleagues return and yourself. So I'll appreciate very much your time, and we'll move on to Dr. Paul Demers.
Thank you.
Cheers.